Mark here is going to be giving a talk on Tuesday, the 27th, Due Diligence and Epistemic Caution, an Impartial Account of Inductive Risk. And then Felipe de Brigard will be giving the annual lecture series on Friday, January 30th, remembering as inverse causal inference. Okay, so now it is my pleasure to introduce.
You have a
Sandy.
cheat sheet for this? Okay, all right, good.
Oh, yes.
Oh, okay.
Now, it's a little bit strange to introduce Sandy since I think everyone knows who she is, but then it occurred to me, not everyone does. So I'm just wondering who's wondering who the hell Sandy. Okay. All right.
Hey,
In some sense, that's true.
well, you're not going to know me in all senses at the end of this thing, I don't think, but you know.
Let me reassure you, you are in the presence now of one of the pillars, one of the mainstays of the HPS community. And now I'm being very serious. It's really a pleasure to introduce Sandy because she really has been someone who's been essential to HPS, and particular Pitt HPS, for many, many years. She goes back to a time before even I was connected with HPS. She was a graduate student here, and then when becoming a member of the faculty, she was chair of the department for a punitive number of years, from 2005 to 2018. How you survived that, I don't know. It was to our immense benefit. She has contributed vastly more to the profession. I'll only mention one thing because I know you want to get on with the talk. She was president of the PSA from 2016 to 2018. I should also mention that her academic work has been a mainstay in the areas in which she worked. Her interests in complex behavior, in multi-level, multi-component complex systems and applications in biology, and of course her work is highly recommended. I know you think I should start winding up, and I will start winding up. She's published extensively. In particular, I'll commend to you her book, Unsimple Truths, Science, Complexity, and Policy, published with the University of Chicago Press, and it was translated into the German, Komplexität, warum wir erst anfangen, die Welt zu verstehen.
Doch.
Yeah, and I don't think that's a very literal translation.
No, actually that came out first. The German came out first.
It came out first.
Yeah.
Okay. And she will speak to us on this topic.
Thank you. Well, thank you all. I really appreciate the opportunity to try out some newish things, you know, built on oldish views, and get your help in where I need to strengthen the arguments, develop the position, etc. So what I'm going to talk to you today about is to propose a kind of pragmatist approach. And just because we had this little exchange, you know, pragmatic is used by a lot of people, and it generally means a kind of means end reasoning that, you know, I look at theories pragmatically, meaning I'm looking at theories for what they're aiming to do and seeing whether they're successful at doing that. Taking a pragmatist view is a different thing, although it may involve kind of means ends reasoning. It's about how we think about what there is, what it does, and how we come to know it. It's really a view about epistemology, metaphysics, and science. And so in that vein, that's the approach I'm going to suggest we explore, or you explore it with me, for understanding the relationships, I think, between metaphysics and scientific practice. And my goal is to be able to, in a sense, read the metaphysics off of the practice, right? As I'll explain, the scientific practice is a kind of modern parlance for things that the American pragmatists took as central, defining, and criterial. And so if you get two pragmatists in a room, they'll disagree about a whole bunch of stuff. So I'm not endorsing, you know, direct perception or any, I mean, I'm not into the crazy stuff, you know, that a lot of the early pragmatists might have adopted, but I think there's a core to what makes something a pragmatist approach. There's a reference at the end to a book that Holly Anderson and I edited a couple of years ago called The Pragmatist Challenge, that tries to explore through the articles of a bunch of people. Some are in this room. Hi, Jim. Thank you for coming. uh, uh, and I have a paper in that book that I'm not going to talk about that content today. So there is a kind of movement. There seems to be a lot of interest in this, and I think part of it comes from the practice turn in philosophy of science that's already a couple of decades old, I think. And that was a move which I kind of, that was the teaser in my blurb, that there has been a desire to move beyond the philosophical analysis of the products of science, the theories, and maybe actual products, things that are that you read about, the kind of intellectual history we do about the ideas as they are published in the articles and the descriptions of theories, the descriptions of experiments, and look at the actual practices that human beings engage in doing science as a source for understanding the issues. And so looking beyond, that was the methodology of science and practice, looking beyond the published works to actual practices. But I want to take that a little step further and say, I want to look at the practices in order to understand what we say. Okay. And so what we believe, what we're warranted to claim is real, what we're warranted to claim is true, and how do we get that warrant through practice? So it's a little bit different, but I think it shares a lot of the motivations of the practice move in science. It does not reject analytical, it doesn't replace the kind of logical, analytical accounts of inference and applying the other kinds of what we might call conceptual engineering, if you know, to the practices of science include theorizing, include developing new mathematics, include all of those things. And so the kind of analytical approaches aren't lost, but they are become part of a bigger story. Okay, so that's the kind of goal. And my target today is to look at biological complexity, surprise, surprise. And I'm going to walk through an example of sociogenetics, which is the study of the genetic underpinning of social behavior. Okay. I want to display how science, how there's a kind of, the picture I'm going to paint, defend might be too strong for what I can do today, but I'm going to paint today, is one in which how in this particular case in sociogenetics, how what science is warranted in claiming is true or real is the output of a kind of continuous iterated negotiation among conceptual engineering of what there is, the kind of ontologies that we posit that are reflected in our concepts and in our representations, including our mathematical representations, and the resources for which provide the resources for representing them, and includes changes in mathematics, theories, descriptions, and the development of new experimental practices and outputs, and in this relationship of empirical and theoretical components of the epistemology of scientific knowledge. Okay, so this is a story with lots of moving parts, so hang on to your seats, right? Okay, so what are the features of pragmatism that I want to pull on for this talk? Is that this is new language, this what's in red. So I'm in love with this week. Let's talk if you don't like it. Pragmatist approach is a method that identifies the evolving equilibria in the theoretical, experimental, and inferential components of embodied and embedded scientific practices that warrant claims about nature. So I want to get to the warranted claims, but I want to get there through this picture of what scientists do, and some of the practices they engage in are a result of, and Edward's not here, so I can talk, I won't talk too much about it, but Edward and I have a grant proposal, we don't know if we're going to get the grant, that focuses on the embodied and embedded components for making good scientific judgments. And so what I mean by embodied is the kinds of both the kinds of equipment we have for reasoning, right, in humans and in our outsourced versions of humans, which may be AI and things like that, but also What's embodied is also what you learn in educational context. That is what is important. Science should be objective. Science should be open. I mean, and then you can see practices immediately relevant to open science or relative to, other peer review, because science should be not idiosyncratic. So you can see there's a relationship between the kind of embedded values, if you like, or norms that we adopt as scientists, qua scientists, and that those epistemic norms contribute to how we make judgments about what's real and what's true. Okay, and so that's an important piece of this story. Um, and um, that's the embodied pace. The embedded scientific practices are also the fact that science is not done in a vacuum. It's not done by empty boxes of inference makers, right? It's done by human beings, and it's not done by them in a vacuum. It's done in a set of increasingly large contexts, including the lab and what the rules of the lab are, the field, what the rules of the norms of the field are, of what we take science to require and the practices associated with that. And again, philosophers have been working on all of these features for a long time, and it is also embedded in larger social, political, cultural contexts that do have an impact on what we study and how we study it. And so understanding this, understanding the object of study has expanded. It expanded with the practice turn in philosophy of science, and I think it expands also with the science and values component of philosophy of science. And as a pragmatist, one of the main lessons from pragmatists is putting humans back into epistemology, right? And I'll get to that in a minute. Okay, so here are a couple of quotes from James and Dewey, because why not, right? And James endorsed what he called a radical empiricism, that the only thing that should be debatable among philosophers should be things definable in terms drawn from experience. And what Dewey interprets experience as practical experience, namely, it has to make a difference in the experience and the behaviors of beings. They don't have to be humans, right? But it has to make a difference. Sellers used to say you needed a difference that made a difference, and otherwise, I'll just drop this in there. Otherwise, it's like James squirrels running around a tree. Okay, it doesn't make a difference what you call it what you want. If it's a difference that makes a difference to experience, then it plays a role in what we should believe and how we should act. Okay, read that as experimental observation, things like that in the scientific context. Dewey on James' view of truth. So what's real are things that make a difference. They're conceptualized things that make a difference to experience. And what Dewey said about James' view of truth statements is that they are made, not a priori. What makes them true is not a priori or eternally in existence, and their value or importance is not static but dynamical and practical. And so that means, again, that truths are made, right? They are not given, okay? So the myth of the given, or the myths of the given, if you work in primate studies. No, sorry. The myth of the given is rejected, that somehow there's a truth out there that's given to us and we come to know it and we do what we can to acquire it. But rather, truth is something that is made in this kind of interaction between cognizers, their representational and perceptual capacities, and things that aren't them, right? Nature, right? Undescribed nature. Okay, so pragmatism in this view rejects scientific absolutism, that there's an eternal truth once and for all, and all we have to do is try to find it. If you're as old as me, you think of Popper's third world, right? Okay, something out there, the true propositions. Where are they? I just need to find them, but rather they are crafted through our conceptual, observational, and other kinds of actors in this kind of dynamics of this view of epistemology. They reject a passive spectator view of knowledge. So you have to put the human back in the story, right? It's not we're outside of nature, we're part of nature, and we're part of making the things true and claim, and the things that we identify as real, right? We are part of that productive process, right? We're not outside of it. And that our beliefs, theories, and representations they reject are not mirrors of nature. So representation stops being this kind of two-place relationship of some form of similarity or, you know, whatever those words are that I can't remember now, about how they map onto correspondence is the general box. It's not about that, what that our representations are just are judged by their being able to be correspond to nature, but rather that our representations are also made by our creating the context in which we can come to an agreement, or what I'm calling an involving equilibria between what we think is real holds up in our conceptual resources, right? Our mathematical representations, our verbal representations, our graphical representations, and our causal interactions with nature through experimentation and observation. How do those all hang together? And so that's the kind of big picture. All right, so that clock doesn't work. Okay, that clock works now. Okay, so so what I want to do with that is, again, so it's a different picture of what scientific knowledge is, what truth is, what real things are. They're part of this ongoing, iterated, changing, but sometimes stable, right, accounts of nature that we as human beings can produce. Okay. So part of this, again, another way to think about it is that we're moving from a thin description of what epistemology is in science to a thicker description. So more things there's a lot of things you have to pay attention to. So it's a set of human practices that shape and support warranted claims of what there is, what is real, and what causes what. And so these are some of the things that go into the thick description of the theoretical experimental practices, that in addition to logical inference, problems, goals, assumptions, activities, representations, instruments, and judgments, all of these are players in this on this stage. And scientific practices are situated, again, in historical, institutional, and social contexts, from individual science all the way up. You can run your nested contexts all the way up to global environments. And global environments make a big difference in certain kinds of scientific practices. Think of the new One Health epidemiological models now that are out there to try to include in their models not just what pathogens do in human populations, but what they do in animal populations and what they're doing in global climate change. So now the global context also can play a role in the establishing what's real, true, and et cetera. Okay, so let's get on with it. So let's go. So I want to take you through an example. How do you do this? Okay, it's a good story. Maybe it's not. I think it's a good story. It's a complicated story. So what are you actually supposed to do, right? So let me take you through some piece of it. And I'm going to look at sociogenetics, which is about the claim that social life in biology, biological social life, I don't know anything about robots. We're not talking about robots here. But biological social life has a biological basis. It's clear. We're not, if you're gonna open wide the thickness of the description, you can't ignore the fact that, social behavior is done by agents who have biological content, right? And is therefore influenced to some extent by genes and the forces of evolution. Okay, so that's just the fact. I mean, if we believe that our current ontology includes genes and our current theories includes evolution, and we think those are well supported and warranted, then it's clear that social life comes under those conditions, and the question is, how does it? Molecular functions of many genes. Okay, so genetics has been the 20th century big story in biology, and it continues to be the big story in biology, but some things have changed, right? So molecular functions of many genes are highly conserved across species, even for complex traits. And what that says is that if you're trying to identify the role of genes in social behavior, right? How do we know what those are? How do we find out what role genes have in the social behaviors? And one of the this is a kind of typical convergence robustness argument, is that if you have the same gene complex in a lot of different organisms who have very similar behaviors, that's some indication that those genes are playing the role in those behaviors. So one of such genes is the gene in the gene complex in Drosophila melanogaster, Apis mellifera, and C. elegans, namely in fly bee and worm. Easier to say. Okay, so there's been studies done in fly bee and worm, and they all share a kind of homologous gene complex called 4. The gene 4 is for foraging, right? So so the gene 4 becomes AMP4 in the honeybee. The gene 4 seems to be associated, right? You get correlation studies in many different technologies here, but is associated with the moving around behavior of these very different organisms for the acquisition of nutrition, right? So that's what it's doing. They're foraging, but they do different things. And the question is, okay, is this the gene for foraging? How does having this gene explain foraging, right? Features of foraging. And the most common features are kind of on-off features. There's more complicated ways of foraging with specialization and things like that. But either you're moving around or you're sitting still, okay? And so what is it that creates that dynamic change, right? If you have the gene, then you respond to certain stimuli and move around. And if you don't, you don't forage, and you don't last very long if you don't forage. Okay, so there's evolutionary accounts of this. I'm not going to do the multi-perspectivalism track here, although we can bring it up at the end. But there's also functional physiology accounts of this. How does it actually work? What are the kinds of processes internal, the mechanisms, the mechanisms internal to the organisms that take the gene 4 and translate it into some molecules, protein molecules, that then interact with these signals, right? And turn on the behavior that moves it or makes it stop moving, right? And it turns out that they're very similar gene complexes in terms of the kinds of amino acid sequences that are coded for in the codons in the gene. We won't go through that whole story either. But for fly and bee, fly and bee and worm, the differences in feeding related behavior in fly, bee, and worm have the same genes, but they don't work it doesn't have the same influence on the behavior in the 3 systems. Okay. So for fly and bee, their differences in behavior are genomic. For bee, they are developmental. Okay, sorry, that should be for fly and worm. I was wondering why this is weird. For fly and worm, they're just genomic. You have the gene, you got it, right? Give me the stimulus, I'm ready to go. That's it, right? But for bee, they are developmental age-related changes in gene expression in the brain. So now what happens is there's a whole bunch of other kind of genetically based biochemistry, as well as epigenetics and social determination happening in this system, and the gene is not going to be have the same explanatory role. What we mean by what is the gene for foraging? Oh, it's the one that turns on and off foraging. Is not a story that plays in all theaters here. Okay, so I'm going to tell you a little bit of the details. But what I want to do first is tell you in general how, so the argument here is, here's an example where I think the ontology of what is the gene for foraging, right? What is a gene? What is a biological basis for this behavior, right? Has changed because what we think of as genes have changed, and varies because different kinds of complex structures recruit it in different ways, okay? So our ontology isn't kind of this, all you have to do is find the gene and you're done with biology, which was, of course, as you know, part of a reductionist methodology, part of a political get the money for the Genome Project methodology. So that view was strong, and unfortunately, I think it's still around in many domains. Okay, so this is to suggest there's more to this description that you need, that we learn by trying to do experiments on what these conceptions of what is real, the real gene is in biology. So here's a little story about the sociogenomic practices. This is going to go kind of quick. And about how the practices, namely the experimental practices, kind of presuppose a certain characterization of the ontology of the gene, right? And when those practices lead to experimental results that fail to accommodate the data that you're getting, get to be revised. And there's a long story. You can start with Mendel, but let's not start with Mendel. Okay, let's start with Mendel for a minute. So Mendel, of course, there was nothing observable in the gene. It was a purely inferential factor. There must be something there that accounts for these patterns of breeding, and all the mechanisms, the tools that he had for instrumentation were differential breeding patterns, and the representations he had were big G, little g factors, right? Okay, so all the representations are tuned to the kinds of experimental discriminations you can make, right, at what part of the organism, and they support a view about what the gene is, okay? But later than Mendel, right, once there was a physical gene, say the early part of the 20th century is the history of the development of the gene concept from this kind of unobservable factor to something locatable on a chromosome to something that could be that was in 53 structurally identified in terms of DNA, right? And later extruded through the process of DNA, RNA replication and translation on a ribosome into a protein that then has to fold and become expressed in the cells of the organism in order for it to do its job. The function of it, hair color, okay, the function of it, foraging, right? Okay, so there's a long story of all of the processes, mechanisms that get you from gene to behavior, and a simple association of the thing that must be responsible for it, i.e. the Mendelian gene, so is no longer plays a big role in biological explanation. Okay, so the initial kinds of experiments included a kind of comparative analysis of actual genetic diversity you could find, because it was hard to know what you're going to do or how are you going to get genetic diversity. You could find wild type genes and study the patterns of their behaviors and how they were different. You could then, and this is Benzer and Beadle and Tatum were the ones who initially identified the most fine-grained association between, in an interventionist way, of how different genes were aligned or correlated with different particular enzymes, right? In mold, I think it was mold in bread or something. Okay, so you could do gene association with natural variations, or you could do single mutations analyses where you just start irradiating flies and see what you get, and you could infer the gene for foraging. I mean, this is our case. They didn't really do foraging there, but you could infer the gene for whatever trait it is you're looking at. But to do that, for that inference to work, you have to assume that there's a simple Mendelian gene trait structure, right? Got the gene, have the trait, right? Don't have the gene, don't have the trait. Well, it turned out that that's what sat behind the practice. Otherwise, the practice makes no sense, right? Okay. And so, but unfortunately, what was discovered is that that structure did not cohere with other experimental and needed new theoretical and ontological development. We have polygenic traits and we have epistasis. We have one trait that has many genes, okay? Okay. And we have one gene that has many traits. Okay, so now you have this many relation, not one relation. And so the kinds of experiments you have to do to study them has to change in light of that kind of ontological update. And so then you get, I'm just giving you moments in this history. And then you get, of course, that, well, okay, so genes work in a network, okay, of multiple genes, right? So what genes in that network have I know it's not just one gene, it's a bunch of genes. What gene in that network plays a role in generating the trait, okay? And then you see the development of new technologies. You get knockout experiments. Now, the knockout experiences were silencing as fine-grained, we have more fine-grained now stuff, but silencing genes and creating double mutants. And now you could look beyond the mold, and these were done in mice, right? You could look at more complex organisms this way. And so the knockout experiments were the next kind of practice that gets developed to cohere with the new understanding of epistasis and polygenic traits. And it was great. I mean, knockout experiments were supposed to tell you if you knock something out and created a double mutant that lived, right? So you could see that it developed its traits, and it was missing a trait, and the ones that weren't knocked out CAD them, then you could say, okay, now that I've used that, I can identify that function of that trait. That's the trait for whatever, right? Fur color or something like that. Now, the trouble is with knockout experiments, and that would be good. And that would be good if, on the assumption that there's a fixed genomic network and there's a genomic network trait relationship, okay? And so if I change something in the network, I should change something in the trait, right? If that guy in the network is playing a role, okay? Now, the trouble is, 30 to 40% of knockout experiments of surviving double mutants were kind of normal, right? Okay, and so the inference structure then was, well, on this assumption, it must be that gene didn't do anything. Okay, I was wrong. Okay, negative result, fine, I've learned. Or I didn't do the experiment right. And those were the two in the scientific community responses because they were holding on to this ontological assumption about the structure of the network. But it turns out there was an alternative view. Ralph Greenspan elegantly presented it in the 70s. And that was that you can't look at networks as fixed structures. So the genome isn't this fixed structure that this always plays the same role and you take it out and it's missing it, right? And that you have a flexible architecture of degenerate genome, degenerate in the, quantum mechanics. I mean, that it has that different nodes in the network, i.e. different genes in this polygenic causal structure, change what they do in the face of perturbation. Okay, so you perturb the network and it doesn't just say ouch and show you what you hurt. It changes its structure. Okay, so this flexible genome is now a whole different ontological space of possibilities for understanding what causes the trait, right? What causes the behavior in this case, all right? And so great, so now we know that we have to look at this flexible and degenerate architecture to know how to, and then again, you develop experimental practices that can detect the features of this new object, right? The genome, the flexible genome. We go from a single gene, single allele, to a genome, to a flexible genome. And now the problem is that too has problems, okay? That too has problems because it turns out that having or not having a gene or a genome, flexible or not, is not sufficient, right, to determine whether you have the trait, okay? Because to have the trait is the result not just of the genome, okay? So it's this presence-absence thing. If I got it, it does it. Well, what's the it? I change what the it is. If it's got it, it does it, right? To, wait a minute, other things are going on. And so one of the things is, of course, in the developmental biochemistry of going from gene to behavior, it turns out that all the cells of an organism, except if they have mutant cancers and somatic things and all that kind of stuff, or if they're chimeras or something, all of the cells in an individual organism have the same DNA. They have the same DNA, but the cells do different jobs, right? Some are bones, some are neurons, some are signal transducers for light in the retina. So all of these things have to be turned on, so to speak. They have to be expressed in the cells in which those proteins need to be interacting with stimulus in order to produce the function that you can read way back to the genome, right? Okay, so it turns out that maybe if you want to know why okay, so let me put up the problem. Why do honeybees forage? Why does they forage and then what and stop? So what is the probability of this behavior? What turns the behavior on and off, right? What's the stimuli? If it's a fixed gene, you just change the stimuli and the gene will respond. The gene's the fixed part. But there's another thing that happens in honeybees, which is they have division of labor, which means that over the lifespan of the individual workers in a colony, they change their biology and they change their behavior. Okay, so young bees are nurse bees. They hang around inside the core of the colony. They feed the baby bees, right? And they tend to the queen who's producing the eggs, laying the eggs, and that's their job. They feed the larva, they tend to there's other kinds of jobs. They could clean stuff up. There's undertaker bees that could take stuff out. Anyway, but they have this, and then as they age, they transition into forager bees. And the foragers are, we're talking weeks long, 3 or 4 weeks as a nurse, and then, 5 weeks as a forager, and then that's it. That was your life. Okay, so anyway, but so there's something that's going on developmentally in the organism, same bee, nurse, forager, but over developmental time, something changes in that bee. And what happens is that there's another question I'm going to ask. Why do the bees at week 4 become foragers and not nurses? Okay, so that's another form of the foraging question, right? And it turns out that, okay, so what's really important is you want to know what the mechanism is that's inducing foraging in the, let's just do from nurse to forager transition. And it turns out that there are important hormonal protein signaling neurological changes that happen. Okay, and some of those changes, so what you want to know is when are the genes for foraging expressed in the individual so they go out and forage? Okay, what makes that happen? So now what you want to do is you want to do experiments not on whether they have the gene or don't have the gene. They have the gene, right? Okay, all their cells have the gene. Okay, all the bees have the gene, right? And so you want to know what is the cause of their foraging when they have the gene, and it means it has to be expressed. Okay, it has to be translated by the RNA to produce the proteins that then generate the interactions that lead to the behavior. Okay, so now what you can do is you want to study, well, let's look at the bees. Let's look at the transcripts, right? What are the RNA transcripts of the protein foraging in the brains of the bees? So these are really interesting experiments, right? And they do them. Bees are actually a pretty good system to study. Okay, so now you want to know, now if you can it's now what the causal agent is the expressed genome in the brain of the bee, and you can detect that through the transcription RNA factors. Does that make sense to people? Okay, so you do transcriptomics. Yay. Now you've got association studies, but now you're associating with something else, right? The RNA transcripts of the flexible genome in the brains of the bees. Okay, so you measure RNA expression of genes in brains that correlates with different behaviors. So now you can see, okay, so if I'm seeing foraging and I'm getting all these RNA transcripts, then those DNAs from which the RNA is transcripted must be causing the foraging, all right? Well, turns out that that's not so good either. And so notice what's happening. We keep changing the phenomenon, changing the causal variables, changing the ontology of genetic causation. And so what you have here is that the difference in transcript abundance was thought to reflect a mechanistic link from gene to behavior. Now, again, we keep changing what the gene is. But now what happens is that transcript abundance is not always predictive of protein abundance. Okay. And remember, it's the proteins that are actually biochemically interacting with stimuli, right? So part of it is light, responding to light, responding to aroma. These are things that moderate foraging behavior, right? Some differences in gene expression are the consequence, not a cause of the behavioral change. So what does that mean? So that means is the genetics of foraging in honeybees is a complex interaction between fixed genetic traits, plastic gene expression, and epigenetic modifications. So epigenetic modifications are things that are not genetic, right? They're epigenetic, right? And they are typically, one version of them is the attachment of other molecules, methyl molecules in methylation, for example, phosphorus and phosphorylation. You attach molecules to the protein after it's been produced by the gene and the RNA and that dance on the ribosome, and now you got your protein, okay? But now you can change what it does. You can regulate what it does by attaching other molecules to it. And that then is inherited cell to cell. So it's a different inheritance form. And it actually then you need to know whether that is inducing foraging behavior or not. Because now the gene by itself, it's not just the gene by itself, it's not just the gene plus all the other genes, it's not just the genes plus all the other genes in their flexible architecture. It's the genes plus all of that and the other molecules that have attached to it post-translational. Okay, so the expression in the RNA, you stop too soon, right, to know what the agency is for the onset of foraging. So one of the things is that it turns out there's a lot of different genetic factors that are related to this, and including some of it has to do, and I won't go, it has effects on, so there's the kind of functional biochemistry of the mechanism of inducing foraging, right? And there's also the DNA methylation as an epigenetic marker, right? That something else is going on. And what that means is that in the brains of the bees, and footnote, in your brains too, right? Things will, from the environment, will change and regulate the gene expression in your brain. And that's what matters to your behavior. Okay. And so one of the things that's interesting is that it's the social environment that actually induces these epigenetic changes that change the role of the gene in the production of this behavior that we're looking at here, foraging. And some very recent work has shown, I don't know what's coming next, do I? No. Oh, here, picture. So thanks to Gemini, which means that things are spelled wrong and who knows what they got right. They put in weird words and stuff. But it was a kind of a good picture about what's going on in this honeybee foraging behavior is epigenetics you see is big, the proteins that actually are expressed, right, which is a function not just of the DNA, RNA, but also of the methylated or the epigenetic factors as well. So one of the recent studies has been done that in the transition from nurse bee to forager bee, you can intervene on that system too, and they have, and you can take out a whole class of bees. Take out all the nurses. Oh no, there are no nurses, right? That was mean. But what it shows is that then what happens when you remove a whole age caste in the honeybee colony, what happens is that the younger bees all of a sudden start developing really fast to fill the nurse gap. And some of the foragers regress and do the opposite of develop, right? I don't know the word. There must be a word for that. I'll have to look it up. But they regress and to take on those nurse jobs, right? Okay, so now it's not even that this kind of developmental program is a kind of linear, absolute, right, unmodifiable piece of mechanistic causation, right? That it itself is moderated by the social environment, by the demographics in this case. There's other cases of social modification, right? In many other organisms, right? Hierarchy structures in fish, for example, change the biochemistry of the fish, right? And so the story is then, I'm going to end. So what is this I'm calling pragmatist metaphysics here going on? Okay, so I'm going to quote myself because, I mean, how many times can you come up with the words? Scientists conceptual and representational practices work in concert with their observational and experimental ones to stabilize the acceptance of a claim of scientific realism. What's the real cause of this foraging behavior, right, in these systems? First of all, there's not just one answer because it's different for worm, bee, and fly. Worm and fly, worms there's not a lot of options for worms, right? Okay, they either forage or they don't, right? And there's more options for Drosophila. There's a little bit more for them, but there's many, many options for social insects like honeybees because the environment is offering many, many more causal variables that are relevant to their very biological causation, the very story of what's the role of the gene in generating this trait. So scientists, all of those things that stabilize for now in this kind of more dynamic picture of what we think what we're warranted to think there is in the world. And the scope of what is deemed real is shaped by our aims and questions. What questions we ask is going to point us to certain parts of the world to try to go through these activities, right? So they make a difference also. But to what we say there is and what we say it does is justified by these ongoing interactions among representative models, causal experience and experiment, and conceptual frameworks in reaching a fallible convergence to what is real, fallible and contingent, right? So there's further features of this pragmatist approach, but this is how you read metaphysics into a story of practice. At least I'm suggesting this is one way we might do it. And read the practices back out from the kinds of assumptions of your metaphysical picture of what there is. You can also, there's also an epistemic argument about perspectivalism or perspectivism that I've run, and I'm running on both the representational forms, whatever your mathematics is, there are things it can't represent, right? Okay, and there's things it can, or whatever your theoretical conceptual framework is, there's things that it gets and things that it ignores, right? And so they're always partial, and in virtue of that perspectival, the things they left in are the perspectives, the things they left out are the abstracted partialities. But that's also true with our causal interactions. If you think of any experiment or intervention or perception, there's things that are attended to, right? I'm looking at you. I'm attending to Wayne. Get the joke? Okay, so there's things that you attend to and things that you don't attend to, right? In any of those causal interactions, there's things that are available to be interacted with in your experimental equipment and things that aren't, et cetera. So those kinds of partialities are also part of the pragmatist rejection of absolutism in all its forms. And it's undermined by there's other things especially in I think everything's evolutionary. I mean, but I'm not going to go toe to toe with a string theorist, although I have tried. The combined open-ended evolution of new forms of representation, certainly we're responsible for those. Think of what mathematics looked like prior to the calculus, prior to other, now we have network representations. I kept trying to figure out how to give you one of those, and the network mathematics of whatever it does. Anyway, okay. New concepts and new means of empirical access. So we get new co-constructed phenomena throughout this development of all of the components. This is to suggest that truths and what is real are made. They're not given or out there. Okay, that's it.
Follow the usual protocol. Let's take a short break for a few minutes. I think there might be more food out there, and then we'll reconvene for questions.
You never know. Okay, thank you for doing this.